It's my distinct pleasure this morning to introduce Dr. Joshua Lang. Josh just arrived this morning from Wisconsin, and his flight was slightly delayed, but he still made it exactly at 9 o'clock as I was about to introduce him. He did his MD at University of Illinois at Chicago in 2006, internal medicine residency, University of Wisconsin, finished in 2008, and an oncology fellowship also at the University of Wisconsin, finishing in 2011. That was 1 year after I finished my fellowship, so I always consider Josh and me to be contemporaries. He's currently associate professor in the Department of Medicine, Division of Hematology Oncology at Wisconsin, and since 2023 has been the associate director for translational research at the Carbone Cancer Center. We talk a lot about physician scientists. I think the term is overused. It's becoming harder and harder for a clinician to also be a scientist these days. But Josh is the real deal. In the field of prostate cancer translational research, he's probably in the top 3 people in the world doing this. I'm not on that list, but I think, Josh, you definitely are. And Josh will, I think, impress you with his research. He has 2 active RO1s. His most recent one is on TROP2 in circulating tumor cells in prostate cancer together with Scott Dame, co-PIs. And a little bird told me that he scored on the 2nd percentile for that RO1. I don't think I've ever known anybody who has had a 2nd percentile, let alone a clinician. He's also a project leader of the recently awarded Wisconsin prostate spore. A little bit jealous of that. And more importantly, Josh is a wonderful human being, a good friend. And as he always does, he puts the patient right at the center of his research. And I don't know what his talk exactly will be about today, but oftentimes he does start with a real patient from his clinic and then gets into some very detailed and intricate science, but always brings it right back to the patient at the end. So with that, it's my great pleasure to introduce my good friend and colleague, Josh.
Doc, is this going to help me? It's the question that one of my patients asked, and I'm going to introduce you to that patient today. And I have to say, I'm just so incredibly grateful for the opportunity to present today. And that, I guarantee, is the kindest introduction I've ever received. So thank you, my friend Emmanuel. So in terms of our discussion today, what we're going to go through is a clinical context for how we understand the biology of treatment-resistant prostate cancer. We'll go through a discussion around liquid biopsies and opportunities to integrate both biomarker and therapeutic development. And then we're going to take a little bit of a transition, take a hard left into modeling metastatic microenvironments and what that means or what it could mean for our patients. So let me introduce you to our patient today. He is a 65-year-old gentleman. So at the time of this clinical encounter, he's presenting with worsening back pain. He has known widespread bone metastatic disease. He's progressed through prior androgen deprivation therapy, an immunotherapy clinical trial, and enzalutamide. However, he received less than 6 months of benefit from enzalutamide, which is very worrisome. So we're talking about what else should we do? We need to make a transition. So when we look at the available FDA-approved therapies for this gentleman, we have the taxane chemotherapies that have been around for over 2 decades. We have, could we consider another AR signaling inhibitor such as abiraterone? Or if he has specific genomic mutations, could we consider a PARP inhibitor? Or newly approved radioligand therapies? And we also have clinical trials. And for the trainees in the audience, the correct answer is always going to be the clinical trial. Now, this is not unique to prostate cancer. This is not unique only to this specific situation. This is something that occurs across all tumor types, across all disease stages. And what it is that when we reach these disease state transitions, how do we make the best decision for the person sitting in front of us today? Yeah, I like to think of it as uncertainty and the art of medicine, of these disease state transitions and decisions at diagnosis. So what did I go to medical school for? It was to do these 3 things. The first is to learn, what do we know about the disease? The next is to understand what's occurring for our patient today. And lastly, what do we know about the available therapies that we can say in this broad spectrum of hundreds of patients, where does my patient fall on that Kaplan-Meier curve so I can make the best decision for that gentleman? So Let's get into it. What do we know about the disease? So prostate cancer is a disease that's incredibly complex, and work led by researchers here at the University of Minnesota and many others across the country, funded by the Prostate Cancer Foundation, said, you know what, this is a disease entity for which we have, our models are not great. So we need to do metastatic biopsies and molecular analysis to understand these drivers of treatment resistance. So this is one of the first and largest, and was my favorite paper in 2015. Where they identified multiple genomic alterations, including molecular alterations, mutations in the androgen receptor pathway, including recurrent hotspot mutations, as well as expression of aberrant splicing variants that could lead to ligand-independent activation of androgen receptor signaling. Other important molecular analysis also identify the alterations in homologous recombination repair genes, such as BRCA2, with a frequency that at that time was estimated about 12.7% of patients. Now, as work has really exploded in this space over the last 10 plus years, we're now identifying subsets of HR mutations that could potentially benefit from new PARP inhibitors, including ones that are with germline alterations that could be inherited and have a critical relevance in terms of identifying patients and family members who should undergo cascade genetic testing. Again, really a truly landmark study that changed how we look at metastatic prostate cancer. Now, at the same time, this cohort of patients has continued to grow over the years, and we're expanding beyond genomic mutations, transcriptional alterations, starting to branch into epigenetic alterations. This is one publication from my colleague, Dr. Felix Feng and George Zhao, who trained with Felix and is now at the University of Wisconsin, where they performed transcriptional profiling of 636 metastatic prostate cancer biopsies. And what they were able to do is classify these different transcriptional signatures, so subtypes of aggressive prostate cancer, using basal and luminal classifications. And they were able to identify these distinct tumor subtypes that also include neuroendocrine differentiation as well. Now, importantly, when they looked at those clinical outcomes, so looking at androgen responsiveness and survival across these different subtypes, what we can identify is that there are a significant portion of patients with luminal prostate cancer that have superior outcomes, especially when we look at treatment with or without androgen receptor signaling inhibitors. It's not that patients with basal cancers don't respond, but the duration of response is inferior. And one of the things that I always like to point out, and I'll show this later, to my trainees, whenever we're looking at Kaplan-Meier curves, I always look at the first 20% that come off of the trial and the last 20%. Who are those patients who have intrinsic resistance, who we need to do better to identify those patients? And then who are those patients that can have long, durable responses to therapy, which itself creates opportunities for precision medicine. Now, that other subtype that I briefly mentioned, that neuroendocrine prostate cancer, or I think in this spectrum of luminal, basal to neuroendocrine and small cell prostate cancer differentiation, those are patients that have, generally speaking, the poorest clinical outcomes. And studies that were done, this is one done at UCSF, where they did biopsies and again, part of the Prostate Cancer Foundation West Coast Dream Team. And what they found was that 18% of patients histologically and transcriptionally could be identified as having a small cell or neuroendocrine transformation. Importantly, P53 and RB loss was often associated or identified in these patients. However, it was not sufficient as a biomarker to guide the identification. We see it in adenocarcinomas as well. Also importantly, from a clinical perspective, is that the site of biopsy itself also did not correlate with these treatment-emergent neuroendocrine prostate cancers. Liver metastatic prostate cancer is always a bad clinical feature. However, many patients with lymph node and bone disease can have neuroendocrine differentiation. And that's led to this really critical concept in treating and understanding prostate cancer known as lineage plasticity. And again, it's this idea that we can have a true adenocarcinoma, but under the selective pressures of our androgen receptor targeted agents, that we can see these acquired molecular alterations, epigenetic, genetic changes that lead to these types of mixed variants or hybrids. And whether that's a double positive, meaning a prostate cancer that expresses the androgen receptor as well as the neuroendocrine markers, a double negative that doesn't express either, or other transitions with these epigenetic reprogramming that lead to a true neuroendocrine small cell phenotype that we can identify histologically, and again, has the poorest clinical outcomes. I think it's really critical from a patient perspective, and this is when I have this discussion of when I'm going to recommend a patient for a biopsy. Here's why: because these small cell prostate cancers, if we find them, can have a tumor doubling rate as fast as every 10 days. I mean, that's a lethal disease in a short number of weeks or months. It's a critical decision point that we need to be able to identify. All right, so let's come back to our patient. So what do we know about our gentleman today? We know his PSA is rising. It's gone from 13.4 to 54 in less than 6 months while treated with enzalutamide. So we do some scans. We have a bone scan performed that shows widespread bone metastatic disease, clearly explains why he has pain at today's clinical visit. So we send him for a biopsy for genetic testing, and we identify a number of mutations very focused around the androgen receptor. So he not only has an AR copy number gain, but interestingly, not only the copy number gain, he also has a 742C missense mutation. He has it at a very high variant allelic fraction. There are some other mutations at lower fractions, but I think we have a pretty good sense for why this gentleman developed resistance to his androgen receptor targeted therapy in such a short period of time. So what do you do with this information? Fortunately, there was a clinical trial that was done for exactly this patient population. So this was the CARD trial published in 2019 in the New England Journal of Medicine. And what they did in this particular study was randomize patients, again, exactly our patient, and randomize them to receive cabazitaxel chemotherapy or another androgen receptor signaling inhibitor. So in his case, he would have received abiraterone on this trial. And importantly, what they found was that the radiographic progression-free survival with cabazitaxel was significantly improved with cabazitaxel versus a second AR inhibitor, 8 months versus 3.7 months with cabazitaxel, and an improvement in overall survival from 13.6 months to 11 months. We saw PSA responses in about a third of patients compared to only 13.5% who received the second AR inhibitor. So this led to the clear indication for cabazitaxel for our gentleman today. But now I'm going to ask you, is this good enough? Now, especially when we look, again, let's ask that question about what are the first 20% of patients that come off the trial? Well, in this case, even in the arm that was superior, cabazitaxel, we saw that more than 20% of patients came off within 3 months, nearly 25% of patients. Those aren't great odds. How about at the tail end? What about those last 20% of patients? Well, you know what? There are some patients that can get durable responses to a second AR inhibitor. It's a very small number, though. And again, that's still occurring within 12 to 18 months. So those are the kinds of discussions that I have with my patients. And it really leads to this concept that while we have many therapeutic options available in prostate cancer, and when you look at this slide back from 2021, I need to update it. It looks like a very crowded landscape. However, cross-resistance is the rule. It's not the exception. So when we collapse down across those classes, what we see is that we do have therapies available. However, that there are not nearly as many as we thought there were or that we hoped would be effective. And it really leads us to, I think, some critical questions in prostate cancer. How do we understand these pathways of progression, these drivers of resistance? So can we identify, again, across diverse mechanisms of resistance, genomic, transcriptomic, epigenomic, others? And if we can identify those, does that create opportunities to provide, I think, what I believe will be a more effective precision medicine strategy for patients? And if we do a good job of it, we'll also improve how we develop therapies for patients as well. So that's going to lead us into a discussion around liquid biopsies. And liquid biopsies, again, sort of this idea that when cancer spreads from one place to another, there has to be some highway that it travels on. Often, not always, but often, it's through blood. So can we find cancer cells using a simple blood draw that will allow us to understand what are those complex molecular alterations? Now, the challenges are when we think about cancer cells or circulating tumor cells, there's roughly about 1 tumor cell for every billion blood cells. So rarity, we're way past the needle in a haystack here, trying to find these rare cancer cells. We also have opportunities to find circulating tumor DNA as well. Again, DNA that's been shed from cancer cells that have lysed or died for whatever reason, and also can be detected in circulation. In projects funded by the Prostate Cancer Foundation, NCI, and the Department of Defense, we here at the University of Wisconsin, here at the University of Minnesota, had collaborations with the good Dr. Dame, as well as others. And this is a publication from our colleague, Dr. Alex Wyatt at the Vancouver Prostate Center. And really, again, established the relevance and validity for looking at liquid biopsies in MCRPC. So in this case, he was looking at circulating tumor DNA and was able to identify that ctDNA content associates with the inferior overall survival. So as a prognostic biomarker, there is relevance to looking at ctDNA. He also demonstrated we can identify AR amplifications, work with Dr. Dames, identified rearrangements, other point mutations. Again, complex androgen receptor alterations can be identified in circulation. We can find some of these other high-risk genomic features too, roughly in about 10 to 20% of patients. So sort of this idea, when we look at liquid biopsies, how can we interrogate this disease biology? And I want to come back to this point. We're going to explore this a little more today of, when I think of ctDNA fraction, ctDNA content, which is moving forward as a potential diagnostic in prostate cancer. We're looking at CTC enumeration. It's really just the tip of the iceberg. We want to do all the things that we do with cell culture in our laboratories using blood samples from patients. And if we can do that effectively, we're going to go far beyond. We're going to explore the rest of that iceberg, looking at gene expression alterations, epigenetic changes, you name it. And that's going to allow us to, I think, rethink how we understand or consider precision medicine in MCRPC. So if we can understand these patient-level mechanisms of resistance, and we can do this longitudinally sampling tumor material, looking from DNA to RNA to protein, I hope we're going to be able to answer these four major questions. Who's going to respond to a therapy? Why does resistance develop? When and how do we target that resistance? And how can we combine or sequence new therapies? So this is how we decided to tackle this. And this is actually going back to my time when I finished my clinical training, and then I went back into the laboratory for 3 years as a postdoc and started working with a collaborator by the name of Dave Beebe at the University of Wisconsin. He's a biomedical engineer who was thinking about these challenges around liquid biopsies. And Dave was taking a very different approach, and kind of different from what I used to think of was micro scale engineering, where you take this really big complex device and you shrink it down into this really small complex device. What I learned from Dave is that all these little tubes are failure points. That's where things can go wrong with not just your technology, but your assay. So what Dave was doing was leveraging physics at the micro scale. We're all in the Midwest. We know in the summers we'll see water bugs sitting on top of the lake. How do they do that? Well, the micro scale, surface tension is a dominant force over gravity. So Dave and his group were using this physics principle to create these micro scale devices that I'm going to show you that essentially leverage this principle of surface tension. And it's sort of this idea that if we can do this well, it's going to be a more simplified micro scale technology that will allow us to do complex molecular analyses. Now, this is the original chip that Dave and his group were developing when I first met them. Actually, it's in clinical trials as a low-cost HIV viral load assay in Africa right now. And the way this technology is designed, what you're seeing here in yellow is an elution buffer, in the middle is a wash buffer, and in red is a complex cell mixture with magnetic particles added to capture our analytes of interest, in this case, whole cells. The clear liquid is oil, so different properties of surface tension. Oil and water don't mix, and within these microscale devices, we can use it to create a barrier. So we apply this external magnetic force, capture our magnetic particles that have bound to our cells of interest, pull them through that first oil barrier, through a wash buffer, through the next oil barrier, and into an elution buffer. So in 45 seconds, we've done a complex cell sorting experiment. We've eliminated any centrifugation or transfer steps, and our original sample is still in place for reinterrogation. So we took this concept and re-engineered this into a vertical orientation that afforded us more operational paths. So what we can do is we can take a patient blood sample, isolate a buffy coat, and put that buffy coat into the device. We can add magnetic beads with antibodies against any surface target that we're interested in. Let them incubate for 30 minutes, apply that magnetic force again, and pull it through that first oil barrier to essentially purify our cells of interest. It's in the next well where we can add antibodies for extracellular staining. Again, let them sit for 30 minutes, apply that magnetic force again, and pull them through that next oil barrier, which effectively removes any unbound antibody. Again, eliminating wash and centrifugation steps. Because if we only start with 10 cells, we're probably going to lose half of them with each of those traditional steps. Now, it's in this next well where we can do imaging for extracellular proteins for single cell phenotyping. We can do fixation, permeabilization, intracellular staining for our targets of interest. We can also add a lysis buffer along with oligo-DT beads, so we can bind mRNA and pull those mRNA-bound beads through the next oil barriers into an elution buffer. Or we can add a high-salt lysis buffer to lyse the nuclei and add silica beads to bind DNA and pull them through the back half of the device to isolate DNA for genomic analysis. So at the end of this, you should have a pretty good sense that I'm greedy. I kind of want it all. But I think if we can evaluate DNA to RNA to protein, it's going to, again, create opportunities for this precision medicine strategies we're hoping to perform. So how does the device initially perform? And this was work published when I was just finishing my postdoc coming out to faculty. And what you're seeing here is what that process is. So we did a series of technology and validation steps. And what we did was we spiked 500 LNCAP cells into 5, 20, or 100 million white blood cells, the equivalent of 10, 20, or 50 mils of blood. And we were able to capture our cells of interest with about 95% efficiency. We then asked, well, how well do we do at purifying our cells of interest? So again, we did that 500 LNCAPs into 5 million white blood cells. And we found that we could reduce the number of white blood cells from 5 million down to about 150. through 1, 2, or 3 oil traverses. And we're able to maintain our capture efficiency around 90 to 95%. We then put 1 cell in 20 million white blood cells, and that green dot we affectionately call the cell, so we got it. So we have single cell sensitivity to find our target cells of interest. And we could do high resolution microscopy for single cell phenotyping. And these are 10 circulating tumor cells from one of my patients with metastatic prostate cancer. With EpCAM in red, the blue is the nuclei, that's Hook stain, and white blood cell markers in green. Now, we've taken this platform and we've moved it forward into multiple different prospective studies, and that's what I'm going to spend the next 15 minutes talking about. So we first asked, using this technology, can we identify, capture circulating tumor cells and perform these multiplex molecular analyses? So this is one of my patients. So he's a 62-year-old gentleman who presented with metastatic prostate cancer. At the time, his PSA was 35.7, again, widespread bone metastatic disease and a retroperitoneal lymphadenopathy and a Gleason 4+5 on biopsy. So he started androgen deprivation therapy and docetaxel chemotherapy. So this was a patient before the Latitude trial was published. He had a great response. His PSA went from 35 down to 0.07 by the time we got to cycle 4. He had stopped all of his opiate pain medications. And what we did was a stain for the androgen receptor. So we did CTC capture and enumeration and intracellular staining for the AR. But what we found at this time when he was responding to therapy was that he only had 6 circulating tumor cells. Now, among those 6, and this is AR nuclear localization on this axis and expression on this axis, but there were still a few cells with very high AR nuclear localization. We then did transcriptional analysis. So we extracted RNA for a targeted panel, and what we found was that, well, there's basically nothing. White is no expression. We saw next to nothing at this time point. So good concordance when he was coming in for cycle 4. What was worrisome is he came in for cycle 5 and his PSA had already started to rise up to 0.42. That is always a poor prognostic sign. So we then, again, looked at his liquid biopsy. We found was an explosion in the number of circulating tumor cells, many with high AR nuclear localization and high AR expression. And for the first time, we're starting to see that ARV7 splice variant that Dr. Dame and Dr. Antonarakis had previously identified and published on. So certainly worrisome, but again, he's feeling fine. And we said, you know what, let's finish your last cycle of chemotherapy and we'll go from there. So he comes in for cycle 6 and his PSA continues to rise. Again, always worrisome. Still a high number of CTCs, but what we're starting to see is increased expression of AR-regulated genes. So his AR axis is becoming more transcriptionally active. Now, after 6 cycles of chemotherapy, everybody deserves a break. So we said, you know what, let's just take a month off. I'm a little worried, but come back in a month and we'll see where we're at and make a decision. And unfortunately, in that month off, his PSA went from 1 to 41. So a dramatic increase in his PSA. And what we saw was that not just did he have a high number of CTCs, not as much nuclear localization when we removed that chemotherapy selective pressure, but now more AR overexpression. But again, now that we see the ARV1 splice variant, and again, his AR regulated genes are blazingly high. So it provides evidence that we can use this type of circulating tumor cell integrated phenotyping and transcriptional profiling to understand or identify mechanisms or progressive disease. So in a collaboration with Dana-Farber, we then did a prospective study. So we enrolled 99 patients with metastatic prostate cancer from UW and Dana-Farber and performed that CTC capture and RNA extraction. And what we were able to identify on the heat map here is there is a clear population of patients with very intense expression of those AR-regulated genes compared to this other group of men here. Now, that transcriptional signature in this particular RT-PCR panel involves PSMA, PSA, NKX 3.1, and TMPRSS2. We also include AR variants given the known prognostic relevance of expression of those genes, as well as the neuroendocrine small cell prostate cancer markers. When we look at this particular cluster, so when we look at what we describe as our AR cluster or cluster 2, what we found is that for patients who are positive for that cluster, that they had a median overall survival of only 8.6 months. Now, contrast that with those who are negative for the signature, where they had a median OS of 22.4 months. We also saw that very similar to what has been published, so that AR variants also were prognostic for survival. So again, good. I felt really good about this data that's consistent with what has been published. When we did the multivariable analysis, we did see that AR cluster was an independent prognostic signature. which leads to lots of other questions. And surprisingly, the ARV itself was not an independent prognostic signature. And as we're delving deeper into this, it's sort of that idea that, well, AR variants themselves can cause resistance and drive resistance to AR signaling inhibitors. So can other AR mutations or alterations that may not ultimately result in an AR variant being expressed. But it really tells us, I think, that we can do these multiplex and look at multiple pathways of resistance in prostate cancer using CTC RNA analysis. So then let's do the trials. So then in collaboration with Mary Ellen Taplin and Raina McKay, we then were involved in 2 prospective phase 2 clinical trials, one with abiraterone and one with enzalutamide. And what we found was that for patients who were positive for that AR cluster signature at baseline, looking at their RPFS, that only 2 patients had an RPFS benefit longer than 6 months, with a median RPFS of 2.7 months versus 41 months for those who are negative for that signature. Now, as we look at that overall survival, again, it still holds out at 15.2 months versus not reached when when follow-up was ended. So what we believe we've identified is not only a prognostic signature, but potentially one that may have predictive value as well. Now, there is an important difference because prediction is for a specific therapy and you need to do multiple prospective trials to ask how well does it perform. So that's where, how do we do that? What do we need to get into those prospective trials? Well, to do that, we need to get into a CLIA certified environment. So in a collaboration, again, great things working with engineers, they build things. So in this case, they built a robot that performs those same processes in an automated fashion that I showed you with that smaller handheld chip. And that's exactly what you're seeing here is this platform technology transferring magnetic particles that have captured cells or captured RNA from well to well for molecular downstream analysis. So we went through this process and we asked, okay, well, how reproducible is the technology, is the assay? So we went through a series of spiking experiments going from 1 to 200 cells and showed nearly perfect linearity using this automated technology. We then asked, well, is it repeatable? So we using different technicians in the laboratory across multiple time points on different days, we then asked, well, how well can we reproduce this finding of cell capture? And it turns out we can do it really well. Again, with the robotic platform, greater than 95% capture efficiency, and it's highly repeatable across different users. Well, what about RNA extraction? So how well do we do with RNA extraction? So what we did was actually develop using, in this case, we used 22 RV1 light cells, did spike in EpCAM capture RNA extraction and found that we could detect ARV7 down to as few as 3 cells with a PCR, a CT value of 31. Now, that's not quite enough in terms of what we were looking for, especially as we're looking at a multiplex, so looking at multiple targets. So we then developed synthetic DNA oligonucleotides for each of our targets of interest and did a series of PCR spike-in experiments and asked, well, when do we start to see dropout? And it's right around 15 transcripts that we start to see dropout for each of the targets that we're interested in. So with this analytical validation showing that we do have very, what we think is very good sensitivity going down to single cell levels and potentially down to 15 to 20 transcripts to detect those particular transcripts of interest. And that's allowed us to define an analytically validated CT cutoff of 31 CQ. Now, how about qualitative precision? So what's that readout going to be? What am I going to do in terms of saying, are you positive or negative for the signature? So, and then how does it reproduce? So, when you draw a tube of blood, well, would we find that sample or those cells in the first tube or the second tube or the third tube? How do we make that decision? So we did exactly that experiment. And again, I want to take a moment to thank our patients at the University of Wisconsin. Over the last decade, we consented over 1,000 patients for blood donation. They allow us to collect up to 50 mils of blood every 4 weeks. We don't do it that often, but it gives us great opportunity and flexibility to do this kind of clinical validation and clinical utility studies. So what we did is we enrolled a group of patients. We took their blood samples and split them into 4 parallel reactions and went through this entire process. And what we found was that for patients who are positive for the signature, that we could identify that in each of those 4 reactions from the same blood draw. And if it was above that analytically validated threshold of 31, every patient was positive at each time. Well, what if patients are negative? Again, it's not only important to identify those patients who are positive, but also that internal validation that those are negative. What we found was that if a patient was negative, they were negative in every single time. Now, the other thing that we did here is you'll see grape IC. So Josh, why are you looking for grapes in your patients? As it turns out, the wine industry is way ahead of us. They actually have developed a synthetic grape oligonucleotide to test the how well is the wine stored? Is it good quality wine? So we actually use that, and we actually do a grape spike-in of a spike-in of a grape oligonucleotide to say, are we doing a good job at capturing our RNA and getting it through this process? So again, internal positive controls. I know, I never thought analytical validation would be so fun. So it's really, we had a blast. Now, what about working with shipped samples? Because again, we're not just doing this here at UW, we want to get samples from across the country. And we did exactly that, where we collected blood samples and processed them immediately versus processing them 24 hours later. And we found, again, if patients were positive for the signature, every sample was positive. If they were negative, every sample was negative. And these patients who are at that intermediate level, again, these would not have been called positive for the assay. So as we're doing this work, we're also thinking, we're talking about adenocarcinomas, luminal basal, identifying those adenocarcinoma features. What about that small cell neuroendocrine cancer that I mentioned before? So we did another study using the same assay, but now again, looking at the expression of synaptophysin and chromogranin A, two transcripts that are classically defined as neuroendocrine small cell cancer markers. We enrolled a cohort of 24 patients at this time. 7 of them had biopsy-proven neuroendocrine or small cell prostate cancer. I can tell you that was a very tough year for me clinically when I had 6 men with small cell prostate. We had 17 patients with biopsy-proven adenocarcinoma MCRPC. They donated blood, and we did this exact assay and looked for expression of those 2 markers. What we found using a single time point that we actually had a sensitivity to specificity, as you look here, a specificity of about 90%, a sensitivity of about 50%, and an accuracy of about 70%. However, when we looked at 2 or more blood samples, we actually increased to 100% sensitivity, specificity, and accuracy, as you're seeing here in the waffle plot here. So an assay that does allow us to identify these molecular alterations or lineage plasticity in MCRPC. So then we jump back into prospective clinical trials, and again, looking at those 2 clinical trials, abiraterone and enzalutamide, well, we found only 3 patients were positive for those gene expression signatures at baseline. But as you can see, those patients did very poorly, and with an overall survival, median OS less than 12 months. So now we have this opportunity to evaluate lineage plastic alterations in MCRPC using liquid biopsies focused on transcriptional alterations. But what is the future? I think the future really is integrating across multiple analytes. It's uncommon for me to say that I don't want more information about my patients. So let's do that exact thing. So here's another gentleman. This is a gentleman who, again, presented with metastatic castration-sensitive prostate cancer, received ADT and docetaxel, and had a great response. His PSA was over 300 and dropped down to 0.3. So we collected a blood sample here at time 0 when he had finished his chemotherapy. And when you see, looking at his heat map here, that he had a little bit of expression of AR, full length and total AR, some NKX3.1, and a little bit of synaptophysin. So a little bit worrisome, but he's feeling great. PSA is very low. And as we continue to follow him, PSA remains very low going at month 3 and month 11. However, at month 14 is when we get a little worried because his PSA goes from 0.3 to 0.7. His oncologist says, you know what, let's have you come back with scans in 3 months because you're otherwise feeling pretty good. What was worrisome when we looked at his CTC transcriptional profile is that not only did we see synaptophysin and chromogranin A, but now NMYC is starting to pop up as well. So he comes back at month 17, and unfortunately, he came back in extremis. He had widespread new liver metastatic disease, widespread bone metastases, and pancytopenia, was admitted to the hospital with a platelet count of 40. He had a biopsy of the liver lesion, and it came back as a pure small cell prostate cancer. He received inpatient chemotherapy, and 2 weeks later, he was discharged as his counts were recovering. And that's actually what you're seeing here at month 18. So when he came back in for his next cycle of chemotherapy, we saw almost a complete eradication of that neuroendocrine small cell signal. Now, importantly, when we did matched ctDNA analysis, we saw that exact same thing. In month 11, the ctDNA content of less than 2%. Month 14, though, up to 30%. And when he started chemotherapy, over 60%. And what we're identifying is PTEN-RB and P53 genomic loss. So very complex molecular alterations that led to the development of small cell prostate cancer for this gentleman. So I believe, again, looking across multiple molecular analytes, I think really in this case was very confirmatory, and I think helps us understand what's driving his disease. Now, I'm really grateful for my collaborator, Dr. George Zhao at UW, and I really want to thank Dr. Marina Sharifi, who's a Prostate Cancer Foundation Young Investigator from a year and a half ago, who asked, can we do more with cell-free DNA? And there's this concept, which I was not familiar with, around fragmentomics, sort of this idea. And I thought that when cancer cells died, DNA was digested. Yeah, we could see some in circulation, but that's about it. Well, as it turns out, if there's protein bound to DNA, that can slow that degradation process enough that we can actually detect that in circulation. So this concept of fragmentomics, can we see differential DNA fragmentation using a blood sample? Now, others had started looking at this exact question. Here is one study looking at patients with melanoma versus healthy donor. And what you're seeing looking at fragment size is a clear difference between those two patient populations. And interestingly, you can also see differential fragmentation at very high ctDNA content, again, like our patient with small cell prostate cancer, but also at very low ctDNA content as well. So again, maybe this might be a more sensitive assay. So we decided to jump into this as well. Now, a lot of folks are working and using low-pass whole genome sequencing, looking at these fragmentation patterns. And some very exciting data has been published by multiple investigators. Alex Wyatt's published in Nature recently. It's a fantastic paper. He has a 300-page supplement. So I don't know what those reviewers did to him, but it wasn't nice. But again, it's a masterpiece. It's really fantastic. And confirmed that we can use fragmentation patterns to identify lineage plasticity in prostate cancer. We can identify meaningful and therapeutic options. Now, there are some limitations, though, is that low-pass whole genome sequencing precludes mutation, alteration detection, and the resolution can be very low, and it's difficult to infer transcriptional activity, again, because we're looking at low-pass sequencing. So we asked the question, could we actually develop a targeted next-generation sequencing panel to try to do fragmentomics, but with specific genes? And in a way that we could potentially integrate somatic mutations, copy numbers, and fragmentation all from the same assay. So we developed an assay. It was an 800 gene targeted ctDNA assay. We enrolled patients at the University of Wisconsin with breast, lung, bladder, prostate, neuroendocrine prostate cancer, a total of 320 patients. And there was also a publicly available data set from GRAIL, so 198 patients with breast, lung, prostate, and then again, healthy donors. This was published in Annals of Oncology in 2023, and what we were able to do is look at all of these panels included, again, targeted panels, exon 1. So rather than the transcriptional start site, which was the more common approach using low-pass whole genome sequencing, we're looking at exon 1 in this case. And what we're able to do is detect those alterations, again, using a Shannon entropy informatic calculation. So we took these two cohorts, we split them into a training and a validation cohort, where George and Marina trained the machine learning model, and then went ahead and optimized that model using a 10-fold cross-validation. And then we assessed performance across our, after having trained and using our validation cohort, we then assessed performance. And what we were able to see was using differential fragmentation, using a targeted panel, we were able to discriminate with a very high AUC between bladder, lung, breast, prostate, and NEPC, with NEPC having an AUC of 1.0 using this targeted ctDNA assay. We had a median ctDNA fraction of 6% in this cohort, which is important, I'll get to in just a moment. And we were able to reproduce these results, again, using the GRAIL cohort as well. Again, maintaining a very high AUC, discriminating normal from lung, breast, and prostate cancer patients. And one thing that I want to point out with the GRAIL cohort is that there's a ctDNA fraction in this group of less than 5%. And Alex Wyatt has published on ctDNA fraction as being incredibly important, and that making mutation calls less than 2%, well, frankly, you just shouldn't do it. It's not going to be accurate, and you could make mistakes for your patients. So we then asked, well, how accurate is this assay as we go down to very low ctDNA fractions, again, to discriminate across different types of cancer? And what we're able to find is that we're able to maintain that AUC going down to a ctDNA fraction between 0.01 and 0.1, and in some cases, less than 0.01. So about 100-fold more sensitive than some of the commercial assays to identify these particularly relevant molecular alterations. And because Scott's here, I have to talk about the androgen receptor. So one of the things we looked at using this targeted panel is, again, we have the AR included, which allows us to look at AR and patient-specific Shannon-1 entropy across exon 1. And what we could see was that the AR does an exceptionally good job across each of these two different cohorts, the UW and GRAIL cohort, to discriminate prostate adenocarcinoma. However, we do see patient-level heterogeneity, and each dot represents an individual patient sample. And as we look at those patients with NEPC, again, we see that internal confirmation that patients with neuroendocrine prostate cancer have low Shannon entropy at exon 1 at exon 1. Again, allowing us to discriminate these two different types of patients, again, with lineage plastic aggressive prostate cancer. In unpublished data, I'm pleased to share that we've now updated this. We've now included kidney cancer. We're also looking at ER positive and ER negative breast cancer, small cell and non-small cell. And again, retaining a very high AUC greater than 0.9 in all cases, though the lung cancer is a little bit of a challenge and we need more patients to really validate and train this model. Now, I did want to take a moment to pause because, as we're doing this work in my laboratory, I'm incredibly grateful. We developed what's our new circulating biomarker core at the University of Wisconsin. It's now an NCI-funded core, part of our Cancer Center core grant. And this group it's a fantastic team of researchers and research specialists, and their job is to process samples from prospective clinical trials, as well as projects that extend outside of my prostate cancer laboratory. So we have 14 multi-site trials. We have 5 more that are currently in development. Last year, well, I guess year and a half ago in 2022, we processed over 650 patient samples and collaborations from 23 different NCI cancer centers. So again, very grateful for this team and this group in terms of what we're able to do moving forward. Now, I want to transition a little bit and talk about how do we integrate these biomarker and therapy development. So I'm very excited about opportunities. And with a recently funded PCF tactical grant with Felix Fang and Pete Nelson, we're trying to ask these questions of how do we understand these kinds of alterations? And can we use circulating tumor cell technology to identify expression of these protein targets of interest with single cell phenotyping or transcriptional profiling and integrate that with drug development as well? So I'm going to focus on one particular target called TROP2 with an R01 that was recently funded to Scott and I. It's a fun story on how we got that grant. We can tell you on the side. But there's drugs out there that target TROP2. One is sacituzumab govitecan. Another is dato-DXd, all in phase 1, phase 2 clinical trials. Sacituzumab or SG is currently FDA approved in breast and bladder cancer. And we asked, and this was a question that Howard Sewell asked me a number of years ago. He came to my lab and said, Josh, it's TROP2 in prostate cancer. Can you find it on CTCs? The short answer is yes. We're able to develop an assay. And what you're looking here is a single prostate cancer CTC with red is TROP2 expression. So it can be densely expressed on the cell surface of these circulating tumor cells. So using our technology, we developed an assay where we captured circulating tumor cells using TROP2, extracted RNA, and did gene expression profiling. And very similar to what we had previously seen using an EpCAM-based capture, was that expression of AR variants and AR-regulated genes was strongly associated with a poor prognosis compared to those patients that even if they had TROP2 CTCs but did not express those AR clusters or ARVs, again, survival was quite different, 7 months versus 14 months. So we think this is a potential therapeutic target in prostate cancer. So let's come back to our patient. So he donated blood. In fact, this gentleman was an Ironman. He donated blood 24 times over 6 years for these types of research studies. And always said, Josh, use these samples to find something to help somebody else. Little did he know that he was actually going to help us find a new drug target for him. So he donated blood, and this is what we got from his CTC results. So in his blood sample, he had over 75 TROP2 positive circulating tumor cells, almost 100 EpCAM CTCs as he's progressing on enzalutamide. His ctDNA content is very high, over 30%. And when we do CTC RNA analysis, we see multiple AR variants, ARV7, V9, high expression of those AR-regulated genes, a little bit of synaptophysin. So he actually is patient number 1 on the clinical trial with sacituzumab. So it was one of the best experiences of my career. I almost said I'm done because he walks in after his day 1 infusion for day 8 and he says, Josh, I stopped all of my morphine. I stopped my pain meds. I feel so much better right now, just after the first infusion. And that was reflected in his liquid biopsies, where we see that his TROP2 CTCs dramatically decreased from 75 to 10 in 1 week and continued to fall over time to almost undetectable levels. His EpCAM CTCs dropped initially, spiked during that time off, the 2 weeks off between therapies, but then we did get a persistent and robust decrease in his EpCAM positive CTCs. His ctDNA content dropped from over 30% down to about 12%. And when we look at his CTC RNA analysis, what we see is that, again, he's positive for AR variants at baseline, but even by day 8, those AR variants are eradicated. So this therapy was incredibly effective against that ARV positive clone. We saw a decrease in expression of those AR-regulated genes as well during his time. However, he ultimately did develop resistance and progressed within about a year on treatment. Now, as we look at his end of treatment sample, there's a little bit of a surprise though, because his AR variants have not recurred. We're seeing expression of AR-regulated genes, so his androgen pathway is active. Maybe a little bit of neuroendocrine prostate cancer, maybe there's some component or some kind of a heterogeneity in his disease. There's something unusual happening for him. So what else could be driving resistance? And that led to this idea of, well, could there be other pathways of resistance that go beyond those types of genomic mutations or tumor-based alterations? We know from work that's come, this is actually not Dr. Felix Feng, this is Eric Feng, his son, and George Zhao published looking at transcriptional profiling across different metastatic sites. And we're able to identify that as we look across lymph node, liver, bone, and lung, that we can see differential transcriptional alteration, expression. So adenocarcinomas and neuroendocrine cancers, as well as differential immune infiltration as well. So could the microenvironment, could the site of disease be playing a role? And again, remembering that our patient has bone metastatic prostate cancer. So when in doubt, come back to your engineers. If you don't have a tool that helps you answer the questions, develop a tool that's going to be more effective. So Dave and his group were working on a particular technology that they call Luminex. So what they do with this platform, this microscale device, is that we create a hydrogel. Whatever cells you want to culture them into this hydrogel, place them in the hydrogel, and as it polymerizes in this microscale device, they actually implanted a PDMS rod through the middle of it. So after the gel polymerizes, they remove the PDMS rod and instill iPSC endothelial cells. And this blows my mind in that those endothelial cells actually self-form in tight junctions with an endothelial line vessel. So now we can actually use this as an approach to actually instill therapies or cells, you name it, into this endothelial line vessel, into whatever microenvironment we want to create. So we decided to tackle bone. So we started culturing all human cells ex vivo to develop osteoblasts, osteoclasts, other stromal cells, monocyte, macrophage populations, as well as prostate cancer spheroids, including ones from cell lines or patient-derived organoids. Using the Luminex device, what we're able to show is culturing these different cells that we could maintain very high viability across all of these different types of organoids. So organoids, macrophage populations, osteoblasts, osteocytes, so viability. And we're now going out to over 2 weeks of time. And kind of interestingly, when we create these microenvironments on a chip, they actually start to support each other. So going out to 3 weeks, we retain greater than 90% viability in these devices. So it creates opportunities to really explore different tumor microenvironments, how cancer behaves, but using that endothelial line vessel, we can also instill therapies. So we went ahead and did exactly that. And these are using confocal microscopy, we can quantify cell death, we can quantify division, we can quantify invasion. So looking at not just DNA, RNA, protein, but now getting it to function. So our first question, well, we're using an antibody drug conjugate with sacituzumab govitecan. So how well does a TROP2 antibody penetrate these different environments? And the good news is when we create these patient-specific environments is that we can get about 2/3 of the drug into these niches. The bad news is that we get 1/3 of the drug does not get into the niche. So there is a drug penetrance issue within bone microenvironments that we're able to quantify now with this platform. We then asked, well, what about creating a prostate cancer that looks like what we see in our patients? So our gentleman today, he is a TROP2 positive, TROP2 high expressing cancer. We have 3 cell lines, LINCAPs, which have very low to no expression of TROP2, D145s, which are kind of intermediate, and LAPC4s, which are high. We cultured them in a bone metastatic environment with osteoblasts, osteoclasts, M2 macrophages, and fibroblasts. And what we found as we using TROP2 low, intermediate, and high expressors, that we have to increase the dose of sacituzumab govitecan in those low TROP2 expressing tumor cells to get any type of cell death. Even those intermediate, however, it's those high expressors, those high TROP2 expressors, even in that bone niche, we do see greater penetrance and greater cytotoxic capabilities. So expression of the target is important. That's not rocket science. But I think that's also where we have to ask ourselves when we're giving these therapies to patients, these targeted therapies, is the target present? We then asked, well, what about the stroma? And I'm going to jump ahead in the interest of time and focus on this slide here. Well, does the stroma have an impact? So we did match culturing of these different experiments. And I will say, these are not high throughput. experiments, when you're culturing 7 different cell types and have to get them confluent and ready to combine together at the same time, not the easiest thing to do. However, when you do it, we're able to do match experiments creating these prostate cancer organoids in the bone chip with or without stroma. And what we see is a dramatic increase across different patient samples, different donors, in terms of TROP2 cytotoxic capacity, including one patient that if there's no stroma, fantastic response. We're only looking at cell culture. However, including the stroma, nearly no cell death at all. So these are potential alternate pathways of resistance and ones that I believe are targetable, especially as we start to identify what is activated bone stroma, or even across any metastatic niche. Can we understand the stromal contribution and find new ways to target those molecular alterations as well? So with that, to summarize, we've developed a CTC RNA assay that is prognostic and is now CLIA certified. We have an upcoming prospective trial led by Andy Armstrong and Dana Rathkoff at Duke and Memorial Sloan Kettering, respectively, funded by NCI and a UH2, UH3 mechanism. So we're going to do the clinical validation study now. That's starting next month. So we're very excited about that. We're now integrating across ctDNA and also really collaborating, because this is something where for some of our trials, we get samples, they say, you know what, we want you to do your CTC analysis, but hold that plasma, we have someone else we want to do it with. Hurts a little, but that's okay. We can send plasma, germline samples wherever our trial sponsors want them to go. We believe there's new opportunities for identifying new therapeutic targets. We talked about TROP2. Again, we are excited about a number of other alterations that are being identified, as well as these new opportunities to identify the non-tumor components and how they may also be driving resistance, but also maybe a new target for combination strategies. Now, with that, I just want to take a moment to thank my many collaborators, and especially those in my lab. Dr. Sperger and Dr. Henninger have been with me for 11 and 9 years, respectively. Dr. Scher, who leads the circulating biomarker core, she's been with me for 8 years. So I'm really deeply grateful for their continued participation in being leaders of this research in my laboratory. The many folks in my lab, as well as collaborators at the State Lab of Hygiene, who helped us get this assay CLIA certified. Our collaborators, this is a list that is growing. And, I need to highlight Scott Dame here, who was my first external collaborator that I had a grant with. We're going on almost 10 years of funding together, of collaborative grants funded. So Scott, good to see you, buddy. Our funding sources, the Prostate Cancer Foundation, my first grant was in the form of a PCFYIA. My second grant was in the form of a Prostate Cancer Foundation Challenge Grant, followed by an R01 and some other DoD grants. And most importantly, our patients and their families. Again, that our patients selflessly donate so we can identify new therapies, new biomarkers. We could not do this without them, so we can really live at that translational space. So with that, I'm happy to take any questions that you might have.
I have microphones for anyone with questions.
Let me just make a first comment and then we'll open it up for questions. So as I anticipated, that was very inspirational, but also very depressing at the same time. And it reminds me of the Charles Dickens best of times and worst of times. We are clearly in the best of times in terms of our technology, but in the worst of times because we are appreciating how complex and heterogeneous cancer and prostate cancer can be. And, I do agree with your multi-analyte method being potentially better than a single biomarker at a time. But as a clinician, if you were going to design a test or a test report, how would you prioritize and rank all these analytes for a given patient, and which one is more important versus less important?
Emmanuel asked the easy questions. So for me, and I think, we've been talking about personalized medicine for over 20 years. And I think the answer to your question is that we need personalized medicine on steroids. That how do we understand, now that we have the capabilities, how do we move forward? And I think this is where I also want to come back to the wet lab. Our discovery biologists who are identifying what the relevance is of these molecular alterations, I think is absolutely vital, because that's how we rank what is the relevance for that patient at that specific time. And I think it's those snapshots in therapy. Where are we seeing, when a patient comes to us today, where is their cancer? And if we don't have a biopsy, and again, great advances in imaging studies, but again, is it giving us the biologic information that we need? It's context dependent. Do we have a therapy for that particular, for what that scan is identifying? So for me, I would always start, we have therapies. So if I have a BRCA2 mutation, I have a drug that targets that. Now, I also want to know, is this a transcriptional signature, a transcriptional driver where the AR is still playing a role in HRR positive prostate cancer? Because in that case, maybe there's an opportunity that we should be doing combination therapies in a way that our clinical trials haven't quite answered for us. Again, because we asked the clinical questions because we had the therapies before we had the biomarkers to do true precision medicine. So I think it's still going to be DNA to RNA to protein. And then how do I prioritize? I think it's going to be based on the therapies that we have available. Because if the protein's there, I'm probably going to do that unless I see some other lineage plastic alteration that will pull me in a different direction. So I think there's ways to do it, Emmanuel, but I think that is the next 5 years that we need to be focused on.
So when you have a patient with advanced heterogeneous disease and you isolate CTCs and then lyse those to get mRNA or DNA, or you take cell-free DNA and analyze that, you're getting a picture of the mosaic of the disease, not a single clone or a single cell snapshot. What's it going to take these liquid biomarker assays and approaches and really start to get to that single cell detail and understand the heterogeneity of the disease and all the individual clones that are participating?
Yeah, so great question, Scott. And I think it's from two different lenses. So from a clinical lens, that single cell capability is less important right now. I want to know what the driver clone of the patient's symptoms at this time point today. And again, if I can really hit that hard, can I identify those residual clones and then find a new way to do combination therapies? And again, great advances in prostate cancer with the use of doublet and now triplet therapies for selected patient populations. Now, can we do that on a truly systematic and patient-specific perspective? Now, at the same time, let's come back to the biology, because the other thing that I said that Emmanuel didn't talk about of what's incredibly depressing is I also said that the liquid biopsies may not be enough, that there's tissue context dependence here. So I've never said we don't ever need to do tissue biopsies again, but I think we need to relook at those biopsies for new therapeutic targets that will help us understand the single cell contribution, but also the microenvironment contribution, which may also explain why immunotherapies have been ineffective in prostate cancer as well. So I think other areas and opportunities for study.
That was a great talk. I'm a breast medical oncologist. So there's so many things that are parallel, but then there are a lot of things that are not. And this is a philosophical question. So in the breast world, a lot of attention is being paid to predictors or prognostic indicators of curable disease, as opposed to metastatic disease. And the idea that we need to, if we're going to impact mortality from the disease, we need to target early stage. And in that regard, and this is not a criticism, it's just an observation of the field, CTCs have fallen out because of the ability to detect CTCs, even with more sophisticated techniques in operable breast cancer, is so low. So I just asking the question, how do you think these approaches go to people you were either operating on, men you're operating on, or radiating with curative intent? Do we need to use this to target adjuvant or neoadjuvant approaches?
Thank you. That's a fantastic question, because cancer, we don't want anyone to have cancer, but if we do, we want it to be curable. And as we've seen in prostate cancer, we really struggle to do that these days with the increased incidence in men presenting with metastatic disease. And I would come back to the biology. So when we're trying to identify, and your point around technologies is really vital, because in some ways, again, we tried to run before we could even crawl, meaning technologies were being spun out saying they could do everything, and they clearly could not. And it took years and years in clinical trials that showed the limitations of those technologies, which also comes back to the importance of analytical validation. Can you do what you say you can do, and can you do it reproducibly and independently that someone else can do that? And that allows us then, I think, to come back and say, in a biologic disease where we see one that is more invasive or is shedding more clones, I think that kind of those initial steps in the metastatic cascade, I think, are really critical for us to come back to. And in those subset of patients where we do see identifiable CTCs with more sensitive approaches, what are those outcomes? Because we expect and hypothesize they will have inferior outcomes and would benefit more. And that's, I think, where it comes also to having orthogonal endpoints, not just looking at ctDNA, and again, going beyond mutation calls, looking at fragmentation patterns. I think that really allows us to push that envelope in terms of, and there's patient-specific assays as well with Natera and others. But now we can push the envelope to say, this is a patient who may have high-risk pathologic features, but one that has low invasive capacity, something that we still struggle to evaluate. Though again, luminal basal kind of gets at that, and breast cancer has been leading the way in transcriptional analysis for that purpose. So I think those are the opportunities where liquid biopsies can complement what we're doing with tissue, but we still need to do the trial.
Really nice talk. had a question with regard to your therapy. We were using an ADC. With the radio therapeutics that are on, what's your opinion of the comparison with those? And for the radiotherapeutics, are they able to go after the CTCs and also your ADC? Are you looking at the susceptibility of those therapies against the CTCs as well as a solid tumor?
Is this still being recorded? So the first part of your question. So we do have new FDA-approved radioligand therapies with Pluvicto. In my experience, Pluvicto has been one of the most disappointing new FDA-approved therapies. I have had 2 out of my last 20 patients get to 6 cycles of therapy. The reason is that we're identifying clonal heterogeneity. We see loss of target expression. We see other lineage plastic alterations. This is research that's ongoing and is actually the highest priority in my laboratory right now, is understanding pathways or drivers of resistance. Because I think we can subset.
This is PSMA, right?
Yeah.
Right.
PSMA targeted radiotherapy.
Right.
Not that there aren't opportunities, and I think it's the challenges we face. And I think, the folks in Australia are getting at some of this using FDG PET and PSMA PET. And is there overlap or heterogeneity? I think that can give us some spatial context. What I still need is the biology. Because if we can understand those biologic drivers, again, is the target present, but is this a cancer that is not sensitive to radiotherapy? Which again, this is also our call to our radiobiologists. This, I think, is an area that is really underexplored of mechanisms of resistance to radio or targeted radiotherapies. So I think that's something else where we need new preclinical models studying this specific question, because it's happening in our patients right now. Now, as you said, that we are able to identify target expression for any cell surface target. We can identify those interactive or drivers of progression. And we're starting to subset some of those out right now. So we're hopeful to have some things to talk more about in the next few months.
And this is a follow-up. So have T-cell engagers been at all useful against, is there a reason T-cells are not particularly effective against this disease?
Yeah, great question. So I think the short answer is there are a subset of patients who respond or benefit from T-cell engagers. There's still a lot of drug development determining what's that optimal combination. So I do think that there is great potential with those types of T-cell engagers, BiTEs, the new triplet or, Ocho bodies. I've heard about those too, creating new ways to combine antibodies. So I think lots of opportunities there. But now we're starting to get into the metastatic niche. And again, I think that's something where we didn't delve into it too much today. But, I started to tell you that sacituzumab govitecan can be influenced by the niche itself. I think that also has really been a driver of resistance to checkpoint inhibitors in prostate cancer. I think it's also, again, potentially driving resistance to some of these new combination immunotherapies. Still its own opportunity, I think, for drug development and diagnostics. But we're not doing it all clinically yet. And I think that's, as I vision for the next 5+ years of prostate cancer, those are the 2 areas that I would prioritize.
All right. We're going to need to move on. Folks we've gone 5 minutes over, so let's give a big thank you to Dr. Lang.